Fantastic. So I'm Brent Stead, CEO of Specific Biologics, and I'm absolutely thrilled to be here today on behalf of the team to present our unique two-site Dualase genome editors, and specifically our first application in repeat expansion diseases. Quick disclaimers, I am going to present forward-looking statements. I am an employee of Specific Biologics and do hold patents related to the work presented today. So, at Specific, we are focused on in vivo genome editing technologies. And the reason that we can do that is, as a platform, our dualases have the ability to do seamless sequence removal, repair, and insertion of both small and large sequences into the genome. Today, I'm going to talk about our focused and differentiated application where we are using that two-site mechanism to precisely collapse DNA repeat expansions. We do have an open $50 million round that we are going to use to advance our AAV Dualase candidate to clinical proof of concept in our first application of collapsing a neurodegenerative repeat expansion disease in a disease area where there are no therapeutics or disease-modifying therapeutics available today. So the in vivo data that I am going to highlight today supports that Dualase is both a safe and durable treatment for this disease. We are directly delivering it to the CNS using clinically and commercially validated AAV. And in this indication, we do believe that we'll be able to get clinical proof of concept through validated biomarkers. As a company, we've been highly capital efficient. We've raised about $11 million to date and have preclinical data in 4 disease areas, as well as data at many other therapeutic relevant targets. Those applications in both sequence removal and repair are catalyzing conversation with potential partners. I'm happy to have a conversation with that through the next few days as well. So our team is also composed of industry leaders who have successfully built and scaled transformative biotech companies. The Dualase technology itself originated from the pioneering work of Dr. David Edgell at Western University. That's also where I completed my training. But before founding Specific Biologics, I spent several years in biologic drug development manufacturing, where I have experience in seeing products from concept into production. And I'm joined to the management team with experts in drug development, manufacturing, and business development who've taken products through clinic and secured successful partnerships. And recently, I'll just mention that Paul Hastings has joined our board of directors, an industry veteran who brings decades of experience in growth and commercialization of biotechs. So together, we really have a team that combines scientific innovation, drug development expertise to really realize the potential of our Dualase medicines as transformative therapeutics. So this is probably the first time that you've heard of Dualase, and what it is designed to do is overcome the key barriers for in vivo genome editing. And we really think about this in 5 key factors. They are efficient at a broad and diverse set of cells and targets. They are precise in that they make the right on-target modification without insertions or deletions or other bystander base changes. They are specific in that we have not seen any off-targets at all target sites tested in cells or in vivo. They're also programmable. As a quick proxy, we've surveyed hundreds of clinically relevant target sites and find that 97% of them can be covered by our current version of Dualases. And particularly important for in vivo genome editing, they are small, only 3.7 kilobases of coding sequence. That's how we package into a single AAV, or we can deliver this as small RNAs. We have built a pipeline of Dualase-based therapeutics where that two-site mechanism has a unique advantage to address the underlying mutation, coupled with clinically and commercially validated delivery vehicles. The initial focus and the subject of today's talk is using that to precisely collapse repeat expansions in a neurodegenerative disease. But we do have in vivo tolerability and efficacy data in other programs and would be happy to connect on those as well. So why are Dualases so special? So as I mentioned earlier, they have 2 domains, and the activity of those 2 domains is coordinated. They are comprised of an engineered variant of an RNA-guided Staph aureus Cas9 domain that is fused to the second nuclease domain called ITEV1. That's the secret sauce of our technology. The ITEV1 domain will bind and cut at a 5-letter sequence that starts with a C and ends with a G, but it will not bind or cut at that sequence unless it's bound to the right site by the Cas domain and properly spaced from the Cas site. So we have an enzyme that has high specificity for 2 sites, hence our name, specific biologics. But importantly, when the ITEV1 domain cuts, it leaves a 2-nucleotide 3' overhang, or a sticky end. When the Cas domain cuts, if we use a version where the Cas domain is active, it can leave a blunt end. And we use those different DNA ends in different ways to bias naturally occurring cell repair pathways to get the right genome editing outcome. We have used DNA and RNA-based repair templates to do both small and large sequence repair and site-specific, including kilobase insertions. And Dualase also removes small and large sequences. That's what we're going to explore in more depth today. And with both of these mechanisms, we can fit that into a single AAV or as all-RNA compositions. So let's look more in depth at that precise sequence removal mechanism. So this actually uses a different format of Dualase than I showed in that first diagram. In this one, we have inactivated the Cas9 domain, but we have active ITEV1 domains. Using 2 guide RNAs that bind just outside of a large sequence insertion, we can orient ITEV1 domains just inside that large insertion. And recall from earlier, the DNA spacing requirement between the Cas domain and the ITEV1 domain dictates which ITEV1 site is targeted. When ITEV1 cuts at the flanking sites within that large sequence insertion, we end up with 2 sticky ends that are compatible. I'm just for illustrative purposes showing here a GG on one side being compatible with a CC on the other. Those ends are substrates for the cell's natural end joining repair pathway, whereby we can remove the large sequence insertion and then resolve those 2 ends precisely. And pathogenic repeat expansions are an ideal target for this strategy because they can be ITEV1 sites over and over and over again. So there we can use 2 dualases that orient ITEV1s just inside the repeat expansion, cut within the repeat, excise the pathogenic repeat regardless of how long it is in a cell or a patient, and then leave a non-pathogenic number of repeats after editing. And we believe this approach is generalizable to many repeat expansions and have done this at 3 repeats so far. So let's look at the cell and animal model data that supports this in our first neurodegenerative indication. So we first assessed Dualase in patient-derived repeat expansion neurons. And I'm showing here a representative differentiation of those cells. We also produce AAV9 that's encoding our Dualase in 2 guide RNAs. That sequence is about 4.6 kilobases from ITR to ITR, and we get good titers out of the AAVs that we're making. We transduce these neurons and can measure editing outcomes. And to do so, we use a method where we can measure the length of the pathogenic repeat versus the collapsed. And here we're seeing 50+% editing across the cells in culture. When we use a GFP reporter, we are actually transducing 50% of those cells, which really highlights the efficiency of Dualase once it gets into the cell. We also assess for off-targets, shown in the graph on the right below, with in silico predicted off-targets and multiplex PCR. We do not observe any off-targets with our Dualase or translocations using this or orthogonal genome-wide methodologies. Dualase is highly specific for its target sequence. To determine the nature of the repeat collapse, we actually sequence that collapse product. In these patient cells, there is an allele that is not expanded with 2 repeat units, and we are measuring the editing of the expanded repeat. We only see 2 outcomes: the 2 unit that is unaffected from the unaffected allele, and then one that has 4 repeat units. So this highlights 2 things: the precision of our system to be able to collapse repeats to 4 repeat units here, and that it's selective for the expanded allele. We also assessed multiple lengths of repeats and see the same 50+% editing efficiency, regardless if it's 160 repeat units or 700 repeat units in these patient cells. As a quick comparison, we did AAV encoding Cas9 using the same guide RNAs that we're using in our Dualase and surveyed for off-targets as well. And we observed a significant off-target in a biologically important sequence. This highlights our specificity advantage of that two-site mechanism where we're using an inactivated Cas9 domain. We do not see off-targets. And finally, in this neuronal cell model, we looked at phenotypic correction, and this manifests in two ways. There's a loss of function where that repeat expansion suppresses a missing enzyme, and a gain of function where that repeat expansion encodes a toxic byproduct. And AAV9 Dualase rescues the expression of the missing enzyme to levels that we see in wild-type cells and also suppresses that toxic byproduct. So we translated this into a humanized repeat expansion mouse model. Using intracerebroventricular injection, we dosed adult 11-week-old mice with 2 doses of AAV Dualase and then sacrificed after 1 month, and then looked at expression of Dualase in the brain region of interest and see nice dose-responsive expression of Dualase. In those same brain regions of interest, we see that that humanized expanded repeat allele is collapsed. And that is also in a dose-responsive manner depending on the dose of AAV Dualase to a non-pathogenic length. And this correlates with a 4 to 6-fold increase in the transcript and protein that's missing for the loss of function. And in those 11-week-old mice, we also looked for the toxic byproduct using a probe for the byproduct and observed that with our AAV Dualase in those brain regions of interest, we reduced that toxic byproduct in adult mice. We're incredibly encouraged by this because it suggests that editing the repeats allows for a natural clearance mechanism of those toxic byproducts. And just quickly, we've done this at other repeat expansions. I'm showing here data from a totally different repeat expansion, totally different cell model. We are seeing 60+% precise editing efficiency in the graph on the left, and that correlates with a 4-fold reduction in a splicing defect that is a hallmark of this disease in the graph on the right. So as a potential therapeutic, we do believe Dualase uniquely can benefit patients with repeat expansions, especially in our first application, neurodegeneration, where we can precisely collapse these expanded repeats to non-pathogenic lengths using single AAVs, which simplifies dosing, manufacturing, and has potential safety benefits, with no off-targets, as we've seen with things like Cas9. And we have the potential to correct the underlying pathobiology of the disease, the DNA repeats themselves, unlocking this as a potential to address 40+ repeat expansion disorders. But we are first focused on advancing this neurodegeneration candidate through to clinical proof of concept. I mentioned that we have a $50 million round open. We have about 50 to 60% of that already through insider and syndicate support with groups in deep diligence now. Happy to connect with anybody on that, of course. That will get us to clinical proof of concept, but we also have an extensive pipeline of other candidates in both repair and other repeat expansion applications, and would be happy to engage with anybody on the conference on those as well. So I hope you join us in our mission of developing in vivo mutation-agnostic genome editing therapeutics, and I thank you for your time and might have a couple minutes for questions. Thanks. Shout out. Yeah, so we haven't disclosed the indication, but there are CSF biomarkers for these diseases. Some of the data that's come out recently from other studies within these disease areas give us some good confidence in the correlation between those biomarkers and disease progression. So we can use those for early readouts in clinics, but our ultimate goal here is to look at functional correction in these patients, of course. That'll be measured over time in the course of the clinical study. Perfect. Thank you.